Hi, this is Jerry Radich from the Fred Hutch in Seattle. And what I'm going to talk to you about are new ways to improve molecular analysis in resource-constrained environments. So when we talk about doing testing in low resource settings, there's a few ways that we have tried in the past, and I'll discuss those and then talk about ways that we're trying to improve on those. So we started doing stuff like this probably about 20 years ago. And at the time, we were thinking that we could maybe import these types of tests that we had developed to other, specialized places in resource-poor settings, universities and the like. But this turned to be terribly tough. The homebrew PCRs are usually, they've been optimized, but they're fickle. They often don't transport well. Some of that's because of kind of local things, rules and the like that haven't been transmitted well into documentation. Some of it's because you need high technical training, expensive machines to kind of pull this off. So the next kind of adjacent solution was developing more user-friendly PCR technology, for instance, the Cepheid cartridge system. And I'll talk about that history just because it's kind of interesting how things worked out. The pro of this is that it's less technical training than standardized reagents. The con is that the machines are still pretty expensive. The cartridges are expensive. Machines break down, et cetera. But it's a pretty robust system. The last kind of most desperate ways is to ship blood to specialized centers. And of course, the advantage there is you have a specialized center that can be doing it. The disadvantage is that it's really expensive to send samples. And samples can be compromised, and you can lose their integrity. It would cost, for instance, from Africa to Seattle, costs roughly $500 to send one tube of blood overnight or 2 days. So that's really not sustainable on a large scale. So I want to go back and kind of give you a kind of a story. It's good to tell stories. This is the Pat Garcia is on the left. She's the head of Max Foundation. She's made an agreement over time that she could give free drug to life to patients in low and middle income countries where she can diagnose be cerebral. Now comes a bunch of really amazing coincidences. Gentleman on my left there, 20 years ago or so, was working with the Max Foundation in Seattle on their computers. He was an ex-Boeing engineer. And Pat was lamenting that she had gotten the Novartis to dedicate free drug for life, which is a fantastically generous thing to do, but she couldn't figure out any way to diagnose these patients first it was Central America and South America. So Bob said, well, you have to go talk to Jerry, because Jerry knows how to do this stuff. The only reason Bob knew that is he was a patient with CML who I actually transplanted 10 years ago. So that's kind of an amazing thing that is hard to explain. The second part of the puzzle came here. Shortly after the 9/11 attacks on New York City, there was actually anthrax attack in the US, sent in these envelopes. Several people died of these attacks. They were sent from a mail station in Hamilton, New Jersey, and then went to outlets at NBC, ABC, CBS, et cetera, including two senators, Senator Daschle and Leahy. And because of this, a company in California called Cepheid started a system of automated cartridge testing for anthrax and other bioterrorism. So Cepheid started off as really a bioterrorism company, and all the mail in the US and following other parts of the world are actually tested by anthrax by Cepheid cartridges before the mail is released. So there was, in the US, we have a holiday called Halloween. We dress up. So because of the run of the fear of anthrax, the US supplies of Cipro basically went away. And so my wife and I on Halloween dressed up as Cipro tablets. Next set of wild circumstances is this gentleman on my left there in gray. Cepheid came to me and talked about wanting to get into the cancer space with their cartridges. So I thought, well, B cerebral would be a fantastic target. The gentleman on the left, Brett Helton, was an engineer at Cepheid at the time, as was his wife. His wife was coming to Seattle to do a PhD in nanotechnology, and Brett didn't have a job. So we convinced Cepheid to put him in the lab, and where he stayed and helped develop the Cepheid automatic cartridge. This is actually my softball team called the Killer Fleas. We were surprisingly good. And this is the result of the Cepheid system, the V3 automated cartridge. And this was really a game changer for testing in the developing world. This is just one example of the machines in Ethiopia and how once we started doing this in the Max Foundation, testing just skyrocketed. The main reason this happened was that the WHO started putting these machines in to test HIV, and TB and the like. And so in these systems, you can insert the cartridge into the machine for any test. And so we sort of ride the coattails of the WHO with a lot of generous support from the Cepheid company, who have been tremendous partners in doing this work. This is some of our outreach work. This is Jordan from our lab, who's also a world-known musician. Apparently he has quite a following in New Zealand. So this is him actually instead of doing music, teaching people how to do the Cepheid system in Uzbekistan. The bottom two panels are all patients who came days to get tested because now they would get free drug. And Jordan was really became the mayor. They took him out to dinner, he went to weddings, and the whole thing. He was quite a hero. And this is where we are now. These are all the Cepheid units in the world. And you can see there's a ton of them out there. So there's a large capacity to touch CML patients for diagnosis and monitoring. But a lot of places can't get these machines still. And so we were working on some ways to advance these diagnostics and make them more available for patients. This is, again, Jordan on the left, and Amy, who was then a postdoc, is now a staff scientist. Actually, she was an engineer, predominantly a microfluidics person before she came to our lab. They've made a little dress out of a World Courier bag. That was a bag that's full of dry ice coming from Africa with one measly tube of blood. So we said, well, there's got to be a better way to do it than this. And so we decided to work on capturing blood samples on paper. It took some time to figure out the right kind of paper. Papers have different pHs and the like. But we kind of stumbled upon finally the right type. So this is except for the protein paper. The paper we ended up using was actually used for protein assays. Quite curiously, it works just fine for preserving RNA. How we proved that we could do this is we teamed up with Sue Bramford in Adelaide. Sue would get us patient sample, run it on Cepheid, immediately blot it on the paper, and then send it to us by snail mail. And the advantage of this is we've kept these things months at room temperature. And so you could imagine a clinic, instead of sending and rushing a sample overnight, could just simply batch a week's samples and send them to us for pennies. And so here's from Adelaide. It took somewhere between 7 to 78 days by snail mail, amazing amount of variations, 68 cents versus 500. And you see Sue does enough good work in Australia that they've actually given her own stamp. This is the XY plot. It's miraculously good. The gray zone is MMR and below. And so if you can just use more spots if you want to get even deeper here. So and here we show that you can actually do behavioral mutation testing on these spots. We've now gone on to basically harvest DNA off these spots as well as RNA. So on CML samples, we can actually do the whole myeloid sequencing panel by using Archer technology. This is an example of getting enough blood to do this just simply by pricking with a diabetic needle. So you don't need necessarily syringes and the like. And this is old. This is, we are now, have done about 1,000 patients on this so-called CML Spot On program from about 40 countries. So it's really been fantastically rewarding work for the gang in the lab. But there's more stuff to be done. As Neil Young would say, rust never sleeps. And so we've been trying to develop, move spot testing to other centers, develop cheaper assays, develop some point of care devices, and look at some acquisition sampling methods. I'll go through those now. This is the world at night. So it tells you again that there's a lot of places that patients aren't going to be able to get testing because there's no electricity. So what to do there? So the WHO has these assured recommendations for devices and LMICs. The A stands for affordable, the S sensitive, the other S specific, U is user friendly, rapid and robust, equipment free, and deliverable to end users. Equipment free means in WHO's no electricity, just the hard part. Now, curiously, so here's a point-of-care device on the right that Cepheid had built. Apparently, they were having a good year because they built one of their cartridge thermocyclers engine, the engine block of the engine. Cool, probably fun to ride, but really not practical to scale up, I don't think. But there are ways to do PCRs without amplification using different enzymes. These are called so-called isothermal reactions. This is one called a LAMP assay, which uses up to 12 different primers to make these complex loop structures. You make so much DNA out of this that you can actually put it under a UV lamp and it will glow. And so this is a device, a self-partitioning device done by my friend and colleague, Dan Chu at the University of Washington. You simply insert amplified through an inlet system on the right here, and by flow through microfluidics, samples kind of self-partition into all these wells, and then you shine a light on it and it glows with this PCR material there. People have done this now for HIV. These are the images for HIV at the bottom, where this is the microscopic image of the amplification, taking the same thing and just putting it in a shoebox and putting it in, recording with a cell phone. And this is a cell phone with a dim light. So you can take these pictures and then send them to the cloud, and then it will use Poisson distribution just to beam you down what the HIV copy number is. So we're starting to work on this now with CML. And how we're doing it is ingenious that my colleague did. He basically, he built these microfluidic devices in the shape of a disc, and then you can mount them on an old CD drive, right? So when CD drives spin to play your favorite tunes, they spin at 1200 RPM and generate 150 Gs, which is kind of amazing. And so that distributes your sample into all of these small cells where they can undergo amplification, either by a thermocycle or by isothermal amplification. At first, we thought we were going to have to actually break the thing and extract the DNA and then recycle it. But it turns out you can just do this and put it in water. This is the amplification plots. And in just 1% water, you get enough amplification to make an end determination of yes and no. Not very quantitative on its own, but when you do the Poisson distributions, this works. And on the right here, these are 3 different ways of doing the same sample. One by RT-PCR, one by Cepheid, one by this machine. You see they're pretty darn close. And so we're trying to gear this up to make a device that will be electricity free, that will be essentially an amplification engine for any sampling target you want, BCR-ABL or anything else. We're also working on point of care, essentially pregnancy tests for translocations. Here we have examples of APL, which would be nice to be able to do quickly at the bedside. And on the far right, CML, these are lateral flow devices where if you put the RNA in, it tracks across by itself without any power. So this is the control band C, and the 12 band here is the PML-RAR. Here that's using just different amounts of input RNA. And then here you've got a gradient where you're looking at control and positives for CML. And so this looks like it's going to work. The hard part is that you have to come with, pure RNA and then do the isothermal cycle, which only takes about 30 minutes, or RPA takes 10 minutes, and LAMP takes 30 minutes. So it's pretty fast, especially for PML RAR and APL. This would be great to be able to do it at the bedside, but you have to get some way to get the reaction stuff in here. So that's the next thing. So we're working with this thing with Irwin here to work on a mechanical leach, which pulls blood in, and this is one that spots on paper. So we have a controlled trial now of this for BCR-ABL versus venipuncture. That trial is pretty much over. The long and short of it is it works, but we're trying to kind of expand it. This is with some other colleagues at the UW, where you draw it up in the leach here, and then you have a stabilizer solution of RNA later. You shake it up, and you basically then can have a preserved sample that's can be then isolated This is an example of a study that was done giving these kits to users because you wanted people to be able to do this at home. So these were patients who or normals who learned how to do this. This is the number of participants, and this is the total number of tries it took them. So it takes most people about 5 to 10 minutes to do this, which is good. Some people here were really fast. This is the pain or discomfort using that mechanical leech. I've done this before. I would say it's almost none. So it's very comfortable to use. The usability of the kit, this is basically people saying that it's very easy to somewhat easy in the vast number of them. And then here's the total RNA yield. It's pretty darn good. And then RNA integrity by the min values is great. And then this is just reference gene expression in PCR. So this thing works. So then the next thing we're trying to do with this is to basically make it so that we pull it not into RNA easy, but actually into a reaction mixture, which you could then just do for 20 minutes and screw on into that little device that the pregnancy test I showed you. You can do it all in one spot. So that's the next kind of task we're doing. Now, another fun thing is looking at ways to amplify DNA, BCR-ABL. As you know, because of the big breakpoints, this is a rather difficult thing to do. It takes multiple PCRs and primers to do it. We thought about doing this a few ways. One is Fluidigm is a system to do microfluidics, where samples get divided into tiny little wells, and then you do PCR on all of them. Again, you basically, it's digital PCR, you detect the BCR-ABL and quantitate it by Poisson distribution. So this has a series of wells and tubes and pumps to distribute samples into as many as 10,000 wells. But one of the things you can do is you can actually make this so that on one side you set up ABL primers, one side you set up BCR primers, and you pump them all into the same tubes, and then distribute the cells across, the DNA across all of them. So each well would get the same DNA, but each well would have different BCR-ABL and ABL primers. So you can then just find out which one's lit up and figure out the breakpoint from that. So that's something that's kind of going on in the background. We also thought about doing multiple plex sequencing, like our friends down the street, Adaptive, where they do VDJ sequencing in ALL by doing hundreds of forwards and backwards primers. Now that took them years to figure out because they had to figure out the concentrations of each of those primers so that you wouldn't have any bias in the amplification. So that sounded like just too much work to us. What we are using now is this nanopore device. This is a device that's about the size of a cell phone. The DNA goes through the system here and actually gets the sequencing done here, and then this plugs into your laptop. So this is an extremely fast device. And how it works is it has a series of biological pores, and you take a DNA, isolated DNA that's not amplified, it's just long read DNA, and it basically separates it into a single strand and pulls it across by its current. And because this thing goes flying across somewhere between 50 to 200 base pairs per second per pore, new technologies have gotten it even faster than that. As this thing goes across this electrically charged membrane, each different base pair deflects the electrical field differently. So you can make the change of calls and bases by this change in electrical field. Your average read is about 150 kD, which is pretty darn big. World record is about 2 million. Need a fair amount of DNA. But when this first started, the error rate per base was about 15%. It's now less than 5% with some of the new technologies and software and the like. But we thought, well, even if it did have a 15% base, if you were just looking to map a breakpoint, like in BCR-ABL, that would be fine. And so the problem is that even if you're going to look at BCR-ABL and say, okay, there's a Philadelphia chromosome of BCR-ABL in every cell, the actual BCR-ABL is just a tiny part of the total DNA. So if you just took pure DNA and ran it across this, 99.999% of the sequence reads would be irrelevant. So what we've done is set up some CRISPR-Cas9 target enrichment where we basically use different guides just to cut out the areas that we're interested in BCR-ABL and ligate these so that, in theory, you're going to be amplifying mostly just BCR-ABL and then not a bunch of other nonsense that you're not interested in. Now, it doesn't work out as perfectly as you'd think. We get about 500-fold enrichment right now. We're hoping to get more, but it helps a bit. And so this is kind of the idea. Here's the BCR, and these, you can't see these arrows, they're pretty tiny, but we've got a bunch of primers there, and for the guides to cut. And so we're kind of enriching this area. And then these are runs where you've gone on BCR, which is backwards. And these are the areas where you actually have gone from BCR and then have broken into ABLE. So these are areas that have both BCR and ABLE. These are ABLE sequences named BCR and ABLE. So this is a patient sample where from this, we could read the BCR-ABLE juxtaposition there. We can develop primers pretty easy. And there it is. There's the read. It's kind of blown up from up there. And then here's our read here, which is the same. So we've done this now in I think about 10 samples. And it's pretty quick. You can do this, with some of the new software that we've been working on, some of our computer scientists. You can get sequence in a day. So this is kind of where we've been going. We do all sorts of weird esoteric stuff in the lab. And so this is one man's opinion on the value of work. This is complexity and cost, and this is clinical utility, benefit, and just feeling good about your work. We've been doing single cell genetics since about 2010. It gives you wild, amazing stuff. Some of it actually might be true. High complexity, hard to know if it's going to have much clinical benefit. Past work we did with prediction and CML is a little bit better than this. BCR-ABLE monitoring clearly is more on the clinical benefit. But I think this global work for myself in the lab has really been the thing that's really driving us right now, because of the amazing amount of benefit we can do from this kind of work. And with that, I'd like to thank all my friends, colleagues, be anxious to talk to you in the discussion section. Thanks.